reach out to the instructor or anybody else. I can help you on the behind the scenes as the administrator. All right, well, I think with that, I'm going to pass it over to one of my colleagues for RN Pharma here today, Elena, who's going to do a quick intro of our instructor, and then we'll kick things off. So thanks again for coming, and Elena, coming to you.
Yes, hello, good morning, and welcome everyone. It is my pleasure to introduce the two speakers. Shannon Pileggi is a lead data scientist at the Prostate Cancer Clinical Trials Consortium, a frequent blogger, and a member of the Our Ladies Global team. She enjoys automating data wrangling and data outputs and making both data insights and learning new material digestible. Daniel Sjoberg is Senior Research Biostatistician at Memorial Sloan Kettering Cancer Center. Daniel was recently awarded the Annual Innovator in Programming Award from the American Statistical Association for his development of the GT Summary R package, a package to prepare data and regression model summaries in ready for publication tables. He is the author of other CRAN packages to perform statistical analysis and to streamline a statistician or data analyst's work, including ggSerfit, dCurves, tidy, CMPRSK, RStudio Pref, Starter, Broom Helpers, and Vizar. So without further ado, the floor is yours.
Thank you so much for that wonderful introduction. I'm going to go ahead and share my screen and get oriented. So as with any online workshop, there is going to be a lot of links and things to juggle. So I want to get you oriented with everything that we're going to be working with. Daniel is going to be helping out monitoring the chat, and Victoria is my colleague at the PCCTC, and she's going to be helping in the chat as well. So it's the resources that you are going to need. We have a website for this workshop where you can find all materials, slides, exercises, and things like that. We're going to be posting that link in the chat. We're also going to be using Slido for this discussion, and we'll kick off those questions in a minute. Please keep that Slido link. It's a little bit easier to group questions and answers together in Slido than in the Zoom chat. So it's a great resource to use. And we're also going to be using RStudio Cloud, and that is going to be posted in the Zoom chat as well. I'll do a few introductory remarks, and then we'll kind of kick off with a little bit of discussion. So I'm going to put my slides in full screen mode. And today we're here to talk about gtreg, a package that Daniel and I developed to produce regulatory tables for clinical research. And Victoria has also been a huge help as an early adopter and giving great feedback on this, and as well as our other colleagues at MSK and PCCTC. So this work is under a Creative Commons license if you would like to reuse or adapt. And again, the instructors of this workshop are myself and Daniel Sjoberg. And if you are going to be working on your own local machine for these exercises, we're going to be assuming that you have a recent R release and that you have a recent version of RStudio. And then the packages, the base packages that you're going to need are gtreg, gtsummary, tidyverse, labeled, and use this. And you want to ensure that you can init your own R markdown files. However, we do also have this wonderful RStudio Cloud workspace setup that has all of the packages that you need in it already. And it also already has an R markdown ready for you to knit with the exercises as well. So I would recommend using RStudio Cloud, but if you want to use your own local computer, you can do that as well. So we are going to be doing a 3-hour workshop today, and we're going to be doing about 50 minutes of work, and we're going to take a 10-minute stretch break, another 50 minutes of work, and then another 10-minute stretch break. And those breaks aren't going to be your activity time. So you're going to have proper exercise time and proper break time as well. And so let's see here, for just a quick overview of gtreg. gtreg is a package that's built on gtsummary, which is also built on GT, which means that these tables that we're going to be producing are highly customizable. But before I get any further into the slides, I want to switch over to Slido. And hopefully you all have the Slido link at this point. And our first question that we have up is what country are you attending from? And I'll wait a few minutes for those answers to come in.
I just put the link in the chat box to Slido, and so you should be able to get that.
Vad kan man inte?
This is awesome. It seems like everyone's getting the hand of managing multiple links and things. So thank you all for contributing your responses. We've got over 40 responses here. And I just love doing online workshops and being able to speak with people all over the world. It is so amazing to see all of you are joining, and hopefully at reasonable hours for yourself as well. All right, I'm going to open up the next question. And this is my first time using Slido, so apologies if anything is a little bumpy. The next question is, what is your native language? As we can see, we have people from all over the world. So I'm expecting a little bit of variety here. I love the diversity that we have in this audience, and thank you all for sharing that with us. So my native language is English, and I am from the South. Sometimes I can have like a Southern twang or a Southern accent. So if at any time I say something that you don't understand, or you want me to repeat myself for any reason at all, just don't hesitate to speak up and ask me to repeat myself or phrase anything a different way. All right, next question. How many years of R programming experience do you have? Oh, that's weird that the Slido bars reorder. It's a little disorienting. Seems like we're really evenly split between different levels of programming experience. So I hope that this package is useful for you no matter what state you are at. That was wonderful. And let's go on to our next question. Have you made adverse event tables in R before? I mean, gtreg is a package that we built to help with clinical reporting in general, but really one of the main drivers for it to get started was for adverse event tables. All right, and so we have a large portion of the audience has actually never made these before. So I hope you can find this workflow useful in your daily work in the case that you do need to make these tables. But we have other types of tables that you can make as well, including standard summary tables and some table listings. All right, so it seems like we have a handful of people that have made those adverse event tables before. And we'll do our last question, just out of curiosity. And it's okay if you've never made adverse event tables. What are challenges you face when making adverse event tables? If you haven't made adverse event tables before, just think about tables in general, any tables you need for your reporting needs. This is an open text response, so I imagine it might take a little bit longer for responses to roll in. So what are some challenges you face when making tables in general? Getting the proper denominator, yes. By group counts. Understanding regulatory requirements, making them pretty, formatting issues, knitting to Word, the statistics, the formatting, factors with 0 cell counts. Okay, standardization, the right denominator, stacking, making them nice, SOC and PT. Journal specific formats. Ooh, that sounds like a tough one too. Labels, tidying. Yeah, so I think these are all challenges that we all know and feel very well. Ordering, yes, all of these things are really, really hard about making tables and also adverse event tables. And so we hope that this package can make your life a little bit easier by taking away some of these really difficult parts, making your workflows a lot easier. So thank you so much for contributing your comments and answers. Keep that Slido open to use it for the Q&A. And Daniel and Victoria will be monitoring that as well. So I'm going to switch back to my slides now. And we're going to talk more in depth about getting started with gtreg. I also have to apologize. I have like a little bit of a tickle in my throat. I really hope I don't cough in your ear, but I'm going to try to mute myself before I start coughing. All right, so here's an overview of the gtreg package. So the gtreg package consists of some functions for tabling adverse events. Those are going to be table AE Count, table AE Focus, and AddOverall. Then we have a set of other functions in the package as well. Table reg summary, table listing, also for making tables, and style XX. So sorry. Style XXX for making table shells. 1 minute. And because gtreg is built upon GT summary, after you make these basic tables, you can then use GT summary modification functions like modify header, modify spanning header, or bolding or italicizing things to get your table looking exactly the way you want. Oh, we're going to make it through this. When you're doing those GT summary modifications, you're going to need to use some GT selectors to specify which columns you want to modify. And lastly, we can use some functions from GT summary to export our output. So let's talk about adverse events. Here's what an adverse event listing might look like. An adverse event, this is a great background for a lot of you that haven't done adverse event tables before. It's a medical issue that occurs during the course of treatment or observation. And adverse events are classified according to a hierarchy. And we're going to typically report lower level terms within a system organ class. So in this case, you can see our lower level term is our adverse event here. And we have our system organ class as blood and lymphatic disorders. Adverse events also record other attributes like severity or grade and likelihood of attribution to treatment. So here we have our grade and our drug attribution. And you notice another feature of our data set is that we have multiple adverse events per subject. So when we go to summarize this table, we have a lot of challenges. I think this was really reiterated in the poll that we just had about finding that correct denominator. So subjects experience multiple adverse events. Thank you for hanging in there with me. I'm going to see what I can do at a break to get this resolved. Not all subjects experience an adverse event. The percent of subjects experiencing specific adverse events is of interest, and typically adverse events are counted by maximum grade per subject. So we have a specific rule by which we count adverse events. And oftentimes when we do reporting, we have to make these adverse event tables over and over again. So we have different requirements like treatment emergent adverse events on treatment cycles, and serious adverse events. All right, I think I'm off mute again. Okay, so when we count by maximum grade, what do I mean by that? So here you can see we have 5 different adverse events of different grades, and some of them are actually the same type of adverse event. So if we separate them out by their lower level term, we have 1, anemia, 2, increased tendency to bruise, and 3, thrombocytopenia. And when we count by maximum grade, that means we're going to count the highest grade per term. So only these 3 terms would get counted out of the 5 terms you see in our adverse event table. And our package is going to take care of that counting for you. Here, this instance showed you counting by maximum grade, but similar logic is going to apply when you count by severity, like mild, moderate, severe. And if that severity is stored as a factor in R, then the highest factor level is retained, which would be severe. So let's talk about your first adverse event tables. The data that come in this package are 2 data sets that we're going to be talking about. We have our DF adverse events, which is our adverse events table. This table has only 10 unique subjects in it, but it's got multiple rows per subject. And the other data set that we have are the patient's data set, DF patient characteristics, which has 100 unique subjects and 1 row per subject, which is a typical setup in clinical trials. Another feature of the data that we have is that our data have variable labels. And if you're not familiar with it, a variable label is an attribute of a variable in a data frame. So where applicable, variable labels and not variable names are printed in your GT summary and gtreg tables. So the example gtreg data comes with variable labels, but you can also create your own via the labeled package. So how you can see your variable labels in our data is if you do stir of a data set, you can see the attribute label here. So here we have a variable name of patient ID, and it's got a variable label of uppercase patient ID. You can also see this when you look at your data set in the viewer pane, and you can see the label printed underneath the variable names. So let's talk about our first adverse event tables using table AE count, table AE, and table AE focus. Sorry, I may have missed this. Is this input a SAS data set? It's actually not. It's actually in the gtreg package. So you'll be able to play with this when we get over to the first exercise, which will be shortly. And thank you all so much for hanging in there. I feel like this is brutal watching me go on mute and cough, and I'm so sorry.
Okay, so we've just finished our discussion of the structure of the data sets. So just very quickly, one is multiple lines, one per patient for one row for every adverse event. And the other one is the patient characteristics, which is one line per patient. So we're going to start by talking about probably the three most commonly used functions in this package at the moment for reporting adverse events. So we're going to talk about TBL AE, TBL AE Count, and TBL AE Focus, and then we'll also review the differences among those three functions. So let's start with TBL AE Count. This one is probably a bit more simple, and maybe you don't need a whole package to help you just count some adverse events because it is that simple, but we will be building upon this example. So here's the code. We take our database, which is one line per patient per adverse event here, DF adverse event, and we pipe that into this function TBL AE Count. We tell it the column that the adverse event is listed in. We tell it the column of the system organ class, and then we tell it also that we want to count, in this example, by grade. So let's take a quick look at what that data looks like. Here's our adverse event on the left, in the middle, the system organ class, and the grade on the right. So again, multiple observations per patient here. So with this, single call to one function, you actually get a lovely formatted table here. You have on the left your system organ class and your adverse events that are indented, and then on the top, on the right, you have grades 1, 2, 3, 4, 5. We'll talk about how to style this table a little bit more to make it a bit cuter in the future, but here we are. There is, with one line of code, a simple adverse event count table, which is something you'll see in nearly every publication about early stage studies, for example, and also in an FDA submission. And by default, where we have 0 counts, we do put dashes or M dashes here, but that can be modified. We do that because oftentimes when I'm reviewing adverse event tables, they'll be very, very large with a lot of different types of adverse events. And it's actually kind of a sparse table because maybe only one patient experienced this type of adverse event or that system organ class. So it kind of eases the eye. It's a little bit easier to read on the eyes in my mind. Okay. So now let's take that a little bit differently from simple counting of adverse events. Now, someone put in there into the chat earlier that it was difficult to identify the correct denominators, and I 100% agree. So let's do TBL AE. So Shannon had mentioned that it's often the case that in adverse event reporting, what you want to do is you want to report the highest grade adverse event per patient that was observed. So if I had, for example, 5 anemia adverse events and 4 of them were grade 1 and 1 of them was grade 3, in a typical table that includes denominators that are the number of patients, you would want to count my 1 anemia adverse event, the grade 3 one, just the one time. So that's what we could do with TBL AE. It's going to add those rates, those denominators for the total number of patients in your study. So to do that, number one, we just use TBL AE instead of TBL AE count, and then we add one more argument to our function called ID. So what that's going to do, it's going to tell the function that this is the patient ID, and therefore within each ID, only count the highest grade, please. So let's take a look at the data real quick. So now we have on the far left, the patient ID. Now this is all patient number 1, and they have a bunch of different types of adverse events and within various system organ classes and at various grades. So you can see here, this increased tendency to bruise This particular example in simulated data has a grade 5 bruise. That's kind of quite severe, but they also have a grade 4. So in this case, only the grade 5 would be counted because it's one patient. So let's take a look at the table. So now we have our counts and our proportions or our rates of those adverse events and those within adverse event and also within system organ class and by grade. You can see here at the top, we have printed n equals 10. So that's to kind of signal to the users that the denominator here is 10. Okay, so and this example speaks a little bit to what I said earlier about using the M dashes where there are unobserved adverse events. It's a little bit easier on the eyes to read something like this. So let's move on to the next example if there are no questions, or there may be a question in the chat.
There's actually a question on the slide, Dan.
Oh, yes.
If you don't have the data in the body systems, such disorder, will these functions still work?
Yeah, so including the system organ class is entirely optional. If you did not include the system organ class argument and just specified the adverse event one, you would get a table that looks nearly identical to this, except for it wouldn't have the rows for the system organ class where the adverse events are indented. They'll just no longer be indented. And so it would operate identically, but just without those system organ class reports. Thank you. That was a great question. Another question. The pipe for the base R pipe can be exchanged with the Magrider pipe. Yes, in this case, that's true. I mean, 100%, they're not 100% exchangeable, but largely, yes, they are exchangeable. So I just want to point out one thing here. One thing that's difficult about these denominators is you have to recalculate the denominator within adverse event and then again within system organ class to get the correct numbers. So this is something that it's just, there's a lot of denominators running around, which is why these tables can be a little bit cumbersome to make, which is one of the lovely things about this package. All right, Shannon, I see you're back on. Do you want to take over?
I don't know if I want to take over. I might let you finish this section and me do the next, but there is one more chat in the table. That was our chat and the thing was a really good eye. Why is there 1 grade 1 iron deficiency, but 0 grade 1 blood and lymphatic system organ class? So that's like a really good catch there.
So I can't see the question. Can you say it one more time?
Yeah. So when we are looking at iron deficiency anemia, we have 1 grade 1.
Yes.
But when we go straight up, we have none grade 1.
Oh, well, within the system organ class. Why do we have 1 iron deficiency, but then that doesn't exist here in the system organ class level? That's a fantastic question.
Yeah.
And it speaks to the difficulty of creating these tables. So imagine I'm a patient who has a grade 1 iron deficiency, but then a grade 3 anemia. So when I'm calculating anemia, I'm going to get included as grade 3. And when I'm counting iron deficiency, you're going to count me as a grade 1. But when I have to do these calculations over the system organ class, which they share, since I can only be counted one time, we're going to account my highest grade adverse event within that system organ class. So in that example, my anemia would be counted and not my iron deficiency because I had a higher grade anemia. Does that answer the question?
Thank you. And yeah, I'm going to try to let my voice come down and try to take over after the first exercise, if that's okay with you.
Oh yeah, of course, of course.
Thank you.
Okay, so one very important part about this table, and one thing that I think is great, is again that we're clearly showing the user that the denominators here are n equals 10, or 10 patients. But I don't know if you remember from a while ago, the structure of the data set was that the data set that was 1 line per patient had actually 100 patients in it. So all these denominators are out of 10, but they're actually 100 patients in our study. So what do we do about that? Well, we add one more argument to our call to TBL AE called IDDF, where we pass the data set that is 1 line per patient so that internally the function can get that data frame and the adverse event data frame, match what the IDs are, and then calculate the proper denominators. So let's just take a look at this now. Again, what our data looks like. So The adverse events portion on the left looks exactly the same, but you can see on the right that there are actually a bunch of patients that may not have appeared in the adverse event database because they didn't experience one. So these are patients 1, 2, 3, 4, 5 who in this case were all on drug B. So let's take a look. How does this table differ from the previous one we saw? So previously we saw that there was one grade 2 blood lymphatic system disorder, and that it was 10% of the data. But now our N is 100, and that has been corrected to 1% instead of 10%. So that internal calculation can be done easily for us, even when there are patients who don't experience adverse events, which is a very common outcome of a study, that at least one patient doesn't experience every adverse event, of course. And to further customize this study, so you saw on the previous slide that patients were in treatment B or treatment A. So oftentimes you have to stratify your reports by the randomization group, for example. So we just add another argument called Strata. So in this case, Strata TRT. TRT is the column in our adverse events dataset and in our patient characteristics dataset that indicates which group that patient has been randomized to. Let's take a quick look at that data again. You've seen this before, except for we're adding on the left the TRT column to DF adverse events. It was already there, but now we're just illustrating it here. And now the table is largely the same, except for it's been stratified by drug A and drug B. You can see that there are still a total of 100 patients, about 44 in drug A group and 56 in drug B. So again, this table is actually complex to accurately tabulate and not fun to do it by hand. And now you really can do it with a single call to a function. That's pretty powerful in my view. All right, TBL AE Focus is not as standard as the other two you would see in like a in an FDA submission, for example, but sometimes I need to know things explicitly, like, did this patient experience any adverse events? Did this patient experience any grade 3 complications or adverse events? Is this adverse event a dose limiting toxicity, yes or no? So these are kind of logicals that are binary, that can be combined across adverse events, across system organ classes sometimes. But let's take a look at what we're working with here. We are going to update our call to the function TBL AE Focus, and largely the arguments are the same, except for I'm going to include 2 tabulations here for did the patient experience any complication, and did they experience a grade 3 complication or or toxicity. So data is exactly the same as what we've seen before, except for each of the adverse events has now been tabulated as any complication, which is going to be true all the way down, and also a grade 3 or greater, which is going to be a mixes of true and false. And here is our table. So again, and at the top here, signaling to you that this is using all 100 patients to calculate these numbers. And we can see here that there are 7 patients who experience any grade complication, toxicity for anemia, and 7 of those were also grade 3 or more. So this is something you'll also see in studies as well. So it's a good one to include. All right, we are to exercise 1. Shannon, do you want to introduce the exercise?
I would love to.
Thank you.
So our recommended method here is to enter the R in Pharma RStudio Cloud Workspace for gtreg. I'm putting my mouse over it, like I can click on it, but you can't. So let's make sure that you have that website handy in the chat. So there it is in the Zoom chat. That's what you want to click on to go to that Cloud Workspace. I will share my screen now. So this, when you go to the workspace, it's going to look like this. Hopefully everyone can get in. There is a gtreg exercises assignment. You can open it up and it's going to deploy that project. And this project should have all of the packages that you need to run these exercises already installed. So you're just going to click over here in your files pane on the gtreg exercises.rmd. And it's going to have some initial code set up for you. So we're going to be, we're demonstrating in our workshop the gtreg data sets. But for the exercises, we're going to have you use some data sets from AddOverall and AddOverall.test. So I'm going to go ahead and run these packages. And I'm going to set a theme from GT Summary to make compact tables. We're going to do a little bit of initial prep on our subject. So I'm just going to go ahead and run this. This is our subject level data. And we're going to do some initial prep on our AE data. So this is our AE level data. And so if you want to kind of get a view of what's going on here, we're going to do glimpse dat subjects. So our subject data is going to have a subject ID, an actual treatment arm, the age of the subject, the sex of the subject, and the race of the subject. And so when we look at our AE subjects, note that we have 158 subjects. And when we look at our when we look at our DAT subjects, we have 158 subjects. Let's do the same for that AE. And so here we have a few more variables. Again, we have our subject ID. We have our system organ class. We have our lower level term. That's going to be the decode here. And we look at it in the viewer pane, you'll be able to see the labels underneath it. So this is our dictionary derived term. We have the severity of that adverse event, whether or not it's a serious adverse event, causality associated with it, and the start date and time, and the actual treatment arm. So just one kind of note that you need to have shared between your DAT AE and DAT subjects is you do need to have the same subject ID, and you do need to have, if you're doing it by Strata, you do need to have the same kind of treatment arm variable that needs to have the same name and the same storage, like character or factor, whatever you're using. So those are the two data sets that you're going to be using for these exercises. And your first exercise is to create an adverse event table saved as T1 that summarizes adverse events by both treatment arm and severity. And be sure to supply IDDF to ensure the correct subject denominator. So I'm going to start the timer and let you all work. You are welcome to keep popping questions in the chat. All right, thank you everyone for working on the exercise. There are a lot of questions and different chats about how things are working here. So first of all, there was one question about, can I download this RMD to my computer? And yes, you can. So you can download this entire project to your computer. And I am forgetting where that download button is. Does anyone online, can anyone online remind me where a user can download this cloud project to their computer?
Yeah, so if you are in the files pane of the RStudio IDE, you click on more to the far right. There's a little down arrow, and then there's export.
Okay. So I'll just repeat that again for anyone that wants to take this over to their own computer. If you're in the files pane, I had to move this pane over to see this option. So you want to move a files pane all the way over. You go down to more, and then there's an export option. And then that will save this entire project down to your computer so you can open this RMD locally on your computer. So that was the first question. Another question that we have is, are there solutions? And yes, there are solutions. I will go over the solutions with you right now. But if you go to the website, there is the same RMD is actually already on the website, and I'll go to the home again and I'll click on it again. So here we have our exercises tab. Go to the exercises. You can also copy and paste this information into your own RMD. And then here's exercise 1. And if you click on this little drop down menu, it'll show you the code solution. So you can go back here and check it along the way to see if your code is correct. And I should have told you that in the beginning, that you could check yourself. But instead of just showing you the code solution, I'll live code it for you, just to make it a little bit more of a better pace. So the prompt was to summarize the adverse events by both treatment arm and severity. We're going to create an object called T1. And so I'm going to take my DAT AE. I'm going to pipe it into the table AE function. I'm going to first do a couple of arguments, and then I'll add a few more to get the complete solution. So we need our subject identifier. So this is going to be use subject ID. We're going to need an SOC term, which is going to be our AE SOC. And we're going to need an AE term, which is our AE decode. And I'm going to do this by severity. So we're summarizing this by severity in this case. I'm also going to put T1 down here so that this table will print in my viewer pane. And this isn't the complete solution. All right. So here what we have is, you can see I have a sub-denominator of n equals 27. And we have our AE summarized by mild, moderate, and severe. But we haven't quite achieved everything that we want to achieve here, because it says supply ADDF to ensure the subject denominator is correct. So I'm going to go in and supply our data frame of IDs. And so our IDDF in this case is from our DAT subjects. And then we're looking here to make sure they have the same subject ID in both. And when we update this table, it showed in line. I think there's a setting you can do it so it shows automatically in the pane. Now you can see our denominator has been updated to 158, representing our subject level denominator. And then our last objective here was also to summarize this by treatment arm. So I'm going to specify Strata equals ACT arm. And then when we pull up our T1, view it over here in our pane. Now we have our AEs summarized by system organ class, adverse event, by strata, and by severity. So that is our complete solution for exercise 1. I think there are a couple of more comments in the Slido. But I think we have, there was one about, I used AE severity and by instead of by strata, and I got similar results. So yeah, you can play around with what you want to, where you want to put different arguments. But this is going to allow you, if you put your treatment arm and your strata and your severity and your by, it's going to allow you to get these nested tables like this. Daniel, I'm going to have you do the slides, and hopefully I can step in for the exercises.
Okay, let me pull it up real quick. Okay. Before we move on, any questions about the adverse event tables we've just created? Okay. So now that we've created these base tables, let's talk about how we could modify these tables. So as Shannon mentioned, there are a number of functions from the GT summary package that you can use to modify the aesthetics of a table. And because a GT reg table meets all the criteria for a GT summary table, all of these functions can be used in this context as well. So in addition to the GT summary functions we're going to use, modify header, modify spanning header, modify footnote, bolding and italicizing labels, there are some GT reg selectors that are going to help us. So those are going to help us select the appropriate columns from these adverse events tables. So the one we're going to use most common is going to be the all AE calls. Is going to pick the adverse event column for us. There's all overall calls, which is going to select the columns that are overalls, and we'll get there in a moment if we haven't already. And then unknown columns, and then there's another one that we won't go over today, I don't believe, called calls in Strata. So if you haven't heard of functions from GT Summary called modify header, modify spanning header, and modify footnote, the modify family is a family of functions in GT Summary that are just used to modify the aesthetics of tables. So as these function names would suggest, you can add footnotes, spanning headers, column headers, and a whole lot of other things. But we're going to focus on these ones here in the upcoming examples. So let's go back to the table that we've already created with using TBL AE, and we will be adding a function called AddOverall. We're going to be adding information with a function called AddOverall. So the AddOverall is actually a generic from GT Summary that is used with all sorts of tables there, but we have written methods for it for our adverse events tables here. And so when you have a stratified adverse event table, we can use the AddOverall function to add columns to get tabulations for both of the randomization groups combined. And then you can see further that we're piping this into bold labels. And what that is going to do is going to bold our system organ class labels, make the table a little bit more aesthetically nice to read. So taking a look at the data, we've seen this all before. We are passing our DF adverse event, which is again 1 line per patient per adverse event. And we're also passing, as the ID data frame, our DF patient characteristics to make sure that our denominators are all correct. So let's take a look here. So you can see here, again, drug A, 44 patients, and we are adding overall patients. Actually, I misspoke earlier. When I said AddOverall added information for both groups combined, that is possible, but by specifying the argument by, what we're doing is we're adding information across the by variable here, which was the grade. So apologies for the misstep here. So we've added 2 columns that have been labeled overall to show us the combined across all grades summaries for drug A and drug B. So, and you can see again on the left that using the bold labels function that the system organ class has been labeled, and it is, in my view, much easier to read with a bolded label there. So, as I alluded to earlier, the AddOverall function has a few different methods for adding overall functions. So you could add essentially a column that looks exactly like drug A, but just put it on the left and that'd be both drug A and drug B combined. You can add these single columns within drug A, one within drug B across the by variables. And there are a couple other options too. So definitely check it out and see which ones are going to meet your needs. So now we want to modify some table headers and spanning headers, and we're going to be using those those selectors that we briefly mentioned previously. So this is a lovely gift that we'll let it walk through a couple times, but we're using the function modify header, and we are changing the primary column label from adverse event to just event. So that column is called label underneath in the data frame, and we will actually discuss ways to be able to figure out what everything is called underneath, so you can accurately specify which columns you want to change. The second step here is using all AE calls to change the labels from just being 1, 2, 3, 4, 5 for the grades. We want it to say grade 1, grade 2, grade 3, grade 4, grade 5. So in that case, we're using all AE calls to grab all of those columns and say we want all of those columns to look like this. And you can see here we're using 2 stars on the outside, so that's markdown syntax to indicate bold. Then we're saying grade, and then in curly brackets by. So when we are using, we're utilizing the glue syntax here. And if you haven't seen glue syntax before, it's super helpful. So essentially, when you see something in curly brackets, we're going to insert that by level right there at that point. The next line in this function, add overall calls, is selecting all the overall columns, which in this case is just that total column. And we are changing it from overall to be total. And there's also, in this case, an unknown column because a grade was unknown. And we're using the all unknown calls selector to change that label from unknown to, it's going to pop up here in a second, unknown grade. I wasn't actually sure what was going to happen there. So similarly, we're using the modify spanning header function to change the spanning header for every single column in those AE calls. And then lastly, there was a footnote update there. So again, these are functions you may have seen from GT Summary already. And because these are GT Summary tables, we can just use them here. So it's a really nice way to be able to use the knowledge you may already have and being able to apply it in other settings as well. Let me just check the chat. Any questions? Not yet. Okay. So here we are, TBL AE with modified headers. So again, let's just walk through this very, very slowly if we can. So we're piping in modify header to the table that we already have. We're saying grab all the adverse event columns, and instead of just saying 1, 2, 3, 4, 5 for grades 1, 2, 3, 4, 5, we want to say grade, and then in curly brackets the by level there. So by, so we will have that as the updated header, and the modify spanning header, we are selecting all of the columns, and we want it to list the stratum level. And also we're using this escape character here to put in a new line, and then below the stratum level, we're going to add the N. So you can see here in the curly brackets, we're adding the stratum level, and we're also adding in curly brackets the N. So those will resolve to the observed stratum level and the observed N within that stratum. So there are 2 important differences between the calls of AE, all AE calls here. So one of them has no arguments, and the second one has arguments true, true. So by default, all AE calls will only select the adverse event columns with the by variable. And when you can specify the arguments true, true, and in this case, the first argument is like, do you want me to select the overall column as well? True. Do you want me to select the unknown columns as well? True. So you can use all AE calls to select every adverse event column, and you'll see how that result is. It's just a slightly different. So you can see here that the new column headers are grade 1, 2, 3, 4, 5. So that's exactly what we wanted. It's perfect. Overall, however, has not been touched because by default, all AE calls is only going to get the by variables. But when we specified that we wanted to update the stratifying headers, or the spanning headers, we said, please include the overall column as well, which is why that spanning header goes over across the grades and the overall column. And you can also see that drug A and the N for the number of patients in drug A's group are now on different lines because we were able to use that, that line breaker \n. So again, this is like really just building on top of each other, and you're getting you can obtain these very customized tables using information or things you may already know from GT Summary as well. So it's pretty powerful in that respect. So that brings us to exercise 2. Hope you kept your RStudio Cloud sessions open. So Shannon, is there anything you'd like me to preface before we get started on exercise 2?
No, exercise 2 is just to modify your table from exercise 1. Don't forget that if you want to see if your solution is working correctly, you can do that on the website, and I can pull that up for you as well. Do you want to keep your computer on, or do you want me to?
I can.
Okay. Thank you. If you feel like you're done a little early and you want to ready to move on, please post in the chat. Or go ahead and take this time to take your break. I know I did promise like 2 breaks and the coughing kind of distracted me a little bit. So take this time as well to take your break and work on the exercise as well. All right, that is time for exercise 2. I hope you're able to successfully execute it. I'll share my screen real quick and walk you through the solution. So we've already created our T1 table. And now we want to modify our T1 table. So we can just take our T1 table and pipe it into further commands. Now, the first objective is to bold the system organ class rows. So in order to bold the system organ class rows, we'll type in bold labels. And now our system organ class rows are bolded. Our next objective is to add an overall column to each treatment arm. So I'm going to do add overall, and I will do across equals 5. And now we've added an overall column underneath each treatment arm. We want to label the overall column as total. So in order to do that, we need to modify the header of the table. And in this case, the header we're talking about is this first row header here. The header on top is going to be the spanning header. So in order to modify the total, we want to do modify header. The columns that we want to modify are all the overall columns, all overall calls. And that's going to touch this column and this column, these two columns. And the way we want to modify it is to have it read total instead of overall. Just modifying the defaults a little bit. And I'm going to put it in double asterisks to bold it. And now we see total instead of overall. And our next objective is in the header to place the number of subjects on a new line within each treatment arm. So to do this, this is going to be the spanning header on top. So we're going to modify spanning header. We're going to use this on all AE calls. And we're going to do this for both any overall columns that we have. So we want overall to be true. And if we had any columns of unknown severity, then we could use unknown to be true as well. But in this case, it doesn't apply. But we'll say true for the overall columns. We're going to modify this such that we have our strata bolded. And we want to see our subject ends on a new line. So in order to achieve that, we're going to do 2 spaces, and then a return carriage in, and then what we want to see in the next line, which is n equals, and then our subject level denominator. Now we have our subject denominators on a new line. And lastly, we're going to add a table caption. And in order to do that, we use modify caption. And we're going to call this our 2014 disorders adverse events. I was hopeful I could keep talking, but my cough keeps coming back. All right, so I apologize for the cough. And now we have successfully modified our table. Please post your questions in the Slido, and I'm going to hand it over to Daniel to keep on with the presentation.
All right, everyone.
There's also one more question in the chat.
What's that?
They want to know how to apply the same format, adding a global overall column across equals strata and renaming it to total with the n number on a new line.
Oh, I don't have my RStudio open to show you at the moment.
I can work on that solution and post it.
Thank you.
Yeah.
All right, so what we've been talking about is primarily some very common ways to report adverse events and new ways to make it very, very simple. But gtreg is not only for adverse event reporting, it does some other things too. So other common tables that you're going to find in a regulatory submission, for example, are going to be just simple patient summaries of like their baseline data, for example. There'll be all sorts of listings as well. And so we have two functions to help with that: TBL reg summary for the more of a regulatory type summary, and TBL listing. So let's talk about those. Or actually, yes, let's talk about it. So the TBL reg summary, and just a callback for those of you who are familiar with the GT summary package, is very similar to the GT summary function TBL summary in that it creates summary tables for the patients when we expect data primarily to be about one line per patient. TBL reg summary is like that as well, but has defaults that are more appropriate in the pharmaceutical realm. So in this case, we are going to be using the dataset DF patient characteristics, which is again one line per patient, 100 patients, summaries of their and has information on their study status and some biomarker and what treatment they are, treatment drug A or drug B. And so we're going to pipe that DF patient characteristics right into TBL reg summary. We're going to specify that we want these results stratified by treatment. And then also in this case, we're going to include only information on markers and study status, and then we're going to bold those labels. So just a quick glimpse at the data. Again, you can see there are 5 patients here, 1 line per patient. All of these patients are in the drug B group. They have these marker values ranging from 4.8 to 7.7 here in these 5 patients that we're looking at. And their status can be that they completed the study, they withdrew, various other things that we'll see in the summary. So again, just pipe that data set straight into TBL reg summary, and you get a lovely formatted table with just like one line of code. And that is incredibly powerful. I love that. I love that I can summarize my data so quickly with a simple call to one function. So here you can see that that biomarker is the first thing being summarized on the left. And because this is a pharmaceutical conference and gtreg is built for the pharmaceutical industry, you can see that rather than like a single row that would just be like median IQR, for example, which is what you'll get in a vanilla TBL summary from GT summary table, we're doing one line per statistic for multiple statistics here. So first reporting the number of observations in each of those, and the mean and the standard deviations on the next row, and the median IQR on the next row, the range on the next row, and the number of missing observations on the last row for that biomarker. And similarly, for the study status, you just have one row per unique study status here. So a bunch of patients completed the studies, a bunch of patients withdrew early for progressive disease, or withdrew for some other reason, or are actively still in this study. Are there any questions on this table? Okay, then we're already to exercise 3. So that wasn't much of a break for your voice, Shannon, but do you want to introduce exercise 3?
Yeah, exercise 3 is to work with table reg summary and add some modifiers to it as well. So go ahead and hit that timer, Dana.
Okay, and let us know if you have any questions. We are here on the Slido chat.
Yeah, if it's the pace is too slow or just right, let us know if you're getting bored or if you want to move faster, or if you'll appreciate the time to play with the package. All right, everyone. I hope this gives you sufficient time to complete the exercise. as well as to answer your amazing questions in the chats. So keep those coming. I really appreciate the discussion, and I really appreciate the downtime also to get to your questions. So for exercise 3, we're creating a descriptive statistics table. We're going to call this T2. This comes only from our subjects data, so we don't need our AE data for this at all. I'm going to remove subject ID so that we don't actually get descriptive statistics on our subject identifier, because by default, table reg summary will produce descriptive statistics for every single variable in your data set. And if you remember, that subjects is fairly small. It's only got a handful of variables in it, and we don't want descriptive statistics on our subject identifier. So we're going to do table reg summary, and we're going to summarize this by actual treatment arm, by ACTA arm. And now we're going to get, and I will type T2 down here so that we can view this table. And so now we have some basic descriptive statistics with our multi-line continuous summary for our continuous variables. To make some modifications on this, we are going to add an overall column. So add overall class equals true. There we go. So now we have our overall column. I can't scoot this over to make it a little bit wider, but here we are. We're going to bold our variable labels to make them pop out. And lastly, we are going to modify our header to make that subject and come up on a different line. So that's going to take a specification like this. So we're going to modify our header. We're going to put in a list because we want to modify 2 different places. So the first thing we're going to do is all stat calls. And we're going to say stat 0 equals false. So we're going to say grab all statistics columns except for our overall column, which corresponds to stat 0 in this case. We're going to specify that we want to bold the level. But to put that in on a new line, we're going to do 2 spaces, a return carriage, and then our in equals in. And you should see that that should do the first 2 treatment arms, but not the overall. So there we go. We have our first 2 treatment arms, have that in on the next line. And then the overall column is just treated slightly differently. So for stat 0, we need to put some explicit behavior there. We're going to do double asterisks on overall, 2 double asterisks close, 2 spaces, return carriage, in equals in, total in this case. And let's run this chunk. All right, so now we have this on a new line. Can we get a little feedback in the chat on, do we need like a proper like 5-minute, 10-minute stretch break, or has the time between exercises been enough for you? So please let us know if you want a proper stretch break or if you want to keep going. Use a break. All right, I got one person that says they can use a break. So let's put on another timer, and I will put one up, and we'll take a proper stretch break, and I'll post the solutions in the chat while we do that. All right, so I believe we are ready to keep going in our slides. Is that correct, Daniel?
All ready to go.
Wonderful.
Okay.
Last hour of the workshop. We got this.
All right. Thanks for sticking with us, everyone. So let's move on to the last function that we're going to talk about. from the package, and that's called TBO listing. So we've talked about adverse event summaries, various ways. We have talked about patient summaries, and now we're going to do listings. So TBO listing is essentially, as the emoji would suggest, it's a fancy way to print grouped or also ungrouped data. So let's first take a look at this code. We are using our adverse events data frame, DF adverse events. We're just taking the first 10 observations because we're doing a listing. So we will keep it simple. We don't want a table that's going to spill over too many pages and too many slides here. We're going to select a handful of columns that we're interested in reporting. Those would be the system organ class, the adverse event, the grade, the attribution of those, and lastly, the patient ID. And we're going to arrange them by adverse event and the grade of that adverse event. And lastly, we are going to print this listing with TBL listing, and we are grouping all of this listing by system organ class. And then lastly, bolding the labels there on line 8. So just a quick look at what the data looks like. This is, what you've seen before, 1 line per patient per adverse event. So this patient is, these first few lines here are all patient 1, and they had various adverse events from various system organ classes at various grades and attributions, which we'll see in the resulting table. So you can see here that it's essentially the same information we just saw on that raw print of the data, but this system organ class has been used as a grouping variable because we specified that grouping in our function argument. And then below that and indented are the individual adverse events along with their grades, their attributions, and the patient ID. So this is a simple way to get an adverse event listing that has been grouped. So there are 2 nice things about this function, I think. First is that it does the automatic grouping by system organ class, which is something that you'll see everywhere in submissions to the FDA and also other regulatory documents. Secondly, essentially what we've done is we've made a generic data frame into a gtreg/GT summary table. And once we make that conversion, you can again use any of the functions that we have already learned about how to modify these tables to modify this resulting table. So this is a nice way to get consistent formatting across all of your tables, adverse event tables, patient summary tables, and listings, and any other data frame that you want to print. So that's, I think, one fantastic thing about this function. I use this function all the time for various results from studies just to report them that I calculated and made in a data frame, and then I'm printing them as a gtreg/GT summary table so that the consistent formatting across all of my tables, making it so cute. Okay, so that's our listing. So you all remember this table, right? With the highlighted rows here. So these have been grouped by term and by system, and we have system organ class and anemia and grade 4. So let's take a look at the code, what we're going to be doing here. So here we will be using that TBL listing, same table we just created. We're going to be grouping by AE label, and we're going to be bolding those labels. And then we're going to be using a really useful function from gtreg and GT summary called asgt. So previously you had these gtreg tables, but we can use the gtreg, the asgt function to convert it to a proper GT function. So GT is a package that is written by Rich Ianen from RStudio, and it is super fantastic and has so many options for styling of tables. So that's what is one of the really strong components of this package, is that you can have fine-grained control over the minute details of these tables using the hundreds of functions exported from GT to style it exactly the way you or your stakeholders may want to see it. So in this example, we are using the GT function tab style to highlight some rows, and in this case we're going to be doing rows 2, 4, and 8. So again, looking at that table, rows 2, 4, and 8, yellow, that's what that hex color is. It looks great. So essentially the takeaway here is that using asgt, you can convert that gtreg or GT summary table to a proper GD table and then utilize the hundreds of functions there to make your table exactly how you'd like it. All right, and that already brings us to exercise 4. So Shannon, you want to give them the lowdown?
Yeah, make a table listing. Open up RStudio Cloud, and we'll see you in a few minutes. Alrighty, I hope you were able to create your first table listing. Let me pop over to my screen, share a solution with you. Okay. And get my things organized. So we're going to create a group table listing of adverse events named T3. What I'm going to first do, just for ease, is I'm going to just filter down to the listings that we want. So I'm going to take my adverse event listings. I'm going to filter on only ones that have probable causality. So that's AEREL equals probable. I'm going to select a few variables of interest that I want to show. So I want to show U sub ID, this SOC, the term, as well as whether or not the event was serious. And I'm going to arrange them in a specific order. So I want to see them listed by a system organ class as well as by the term. And so when we look at our dot list, you can see here there's our variables and just a handful, 16 observations and our listings here. So now I'm going to create my table. I'm going to take my dot list. I'm going to pipe that into table listing. I'm going to group by the SOC term. And then I'm just going to bold those labels to make them pop. Oops. And then print it out when we're done. So let's see our T3. And here we have our grouped listings. So you can see our different adverse events reported underneath different system organ classes. All right, I think I'm going to let Daniel take it away to the next section. Unless there's any questions about table listing that we need to address.
All right, let me share my screen once again. All right. So if you've been involved in the creation of adverse event summaries and other types of submissions for regulatory purposes, you are going to be intimately familiar with table shells and how annoying a table shell is to create. One lovely thing about this package is that we've kind of married the creation of the adverse event summaries and the patient summaries and the creation of the table shells. And by doing that, we make sure that it's actually very easy to create the precise table outlined in the table shell because they were generated from the exact same code. So let's go over what we're going to be using for this section of the presentation. So we're going to be using a function called style XXX. So if you are already familiar with some of the GT summary functions like style number, it's essentially very similar to the scales version of formatting numbers function, except for it has some ways to control it in a GT summary way. So, and they have style sigfig and style ratio for formatting ratios. So here we have style XXX. It's not as scandalous as it sounds. So don't worry. But what it's going to do is going to, instead of like rounding integers or other variables to, or other numbers to like 1 or 2 decimal places, it's going to essentially just give us X's for table shells. So there's an entire vignette about this. So check out the link at the top. It has a lot more examples and it'll be super helpful to go over more of the complex cases. Here we're going to cover some of the more straightforward ones. So in order to create this table shell, we're going to create some dummy data, or we can just use any data that we already have collected from adverse events, for example. We're going to pass that data to the function of our choice. I think our example here is going to be TBL AE. And then instead of showing the actual statistics, we're going to override how those statistics are formatted using style XXS to replace those numbers with X's. So a quick little example of how this works. So style XXS, if we pass it a sequence of integers from 8 to 11, by default, it's just going to give us 2 X's back for every single thing. Similarly, we can add the width and digits arguments. So a total width of 4 characters and 1 decimal place, so 1 digit. And now that same code would return xx.x to indicate that, oh, in these table shells, there will be a decimal place that's shown. I see a couple of questions in the chat. Is there anything we should address before we moved on? Oh, yes, safe for work. Okay, so here we are going to be, going back to our favorite old function here, TBL AE, and most of the syntax is exactly what you've already seen, but we're going to be adding a digits argument. So digits is going to specify how the statistics are rounded and formatted typically, but when you're in the mode of creating table shells, we're just going to pass the style XXS function so that so that these tables will have X's instead of actual statistics. And we're going to set the 0 symbol to null, because in this case, we don't want to put an M dash where there's unobserved data. We really just want X's everywhere. So that is the default, to put that 0 symbol back to null so that it would show the zeros in general. But in this case, we're going to be using style XXS to replace those numbers with X's. And at the bottom here, we're again using modify spanning header to specify the stratum levels, and also just putting X's for the number of observations within each stratum. So that's the code. So just 2 small modifications essentially to what the code we've already seen. And look at this cute table. So many X's, such a gorgeous little table shell for us. And what I, again, really appreciate about this table is that because we were able to use TBL AE to create the table shell, it's going to be incredibly easy to use TBL AE to make the actual table when the data arrives in your email box, or in a secure server that you connect to, I should say. So, and then the data, we have seen this before, so I won't focus on this much too at the moment. All right, and we have an example or exercise. Shannon, do you want to give a summary?
I think you did skip over one slide if you want to go back one.
Whoops.
Yeah, because what we talked about a minute ago was
which one was
it? We did a uniform shell. So what if you want all of the numeric values to display the same, like only 2 X's? But there might be instances in which you want some numeric values represented by decimal place, and I call that a custom shell. So go back and then whoop, whoop, whoop.
Where am I going? What have I done here?
All right, one more. Go one more slide.
One more, frontwards or backwards?
Forwards. And now we have a custom shell. Click on that code over there instead of the data tab.
Ah, I see. I thought I was advancing to the data tab on accident. Okay, right? I see.
Yeah.
All right. Save your voice, Shannon. I'll discuss. So here we're going to be using again TBL AE, but instead of showing 2 X's for every single statistic, we want to show that we're going to be reporting the percentages to 1 decimal place. So instead of just passing the 1 function to the digits argument, style XXS, we pass a list of 2 elements. The first is just style XXS, XXX, and the second is another function, but specifying that width 4 and 1 digit argument to style XXX so that we can specify that. So essentially, the list is of a length 2. We're passing 2 functions, and the first argument is going to say how the n is reported, and the second argument is going to indicate how the percentage is reported. So because there are 2 statistics reported, we pass a list of 2. So that's how that works. Thank you for stopping me, Shannon. So here is the table you saw previously, except for, you can see here, The numbers, again, just XX, but the proportions, the percentages here are now XX.X. So they're highly customizable, and you can get whatever kind of table shell you need. All right, thank you. Thank you.
Yeah. There's also a question in the chat, like if we don't have the data yet, what do we do? So we would create some dummy data in that instance. And on the Slido link, I linked to the vignette on gtreg, which will show you how to do that in some cases. Yeah, and we can post that in the Zoom chat if you're not on the Slido too. So we are going to go to exercise 5 now, which is to create a table shell. Exercise 5 actually builds on top of a previous exercise. So I'd recommend kind of grabbing some of that code. I'm going to cough, so I'm going to go off and let you start. All right, I hope you were able to successfully create a table shell. Going to pull up RStudio Cloud. I'm going to go back up and grab my T2 exercise where we created our summary statistics table. I'm going to call this T2 shell instead and make a few modifications. So the first modification that we want to make in table reg summary is we want to specify how we want our digits to look. And so we're going to do this for everything in this table. We're just going to take that uniform approach instead of the custom approach. And what we're doing here is we're actually selecting which variables this applies to. So you could apply your styling differently to continuous or categorical variables, but here we're applying it to everything. So I'm going to modify that T2 shell. And now we have our X's applied for all numeric values in the table. And you see here that we still need to do this in our header as well. So instead of having our in included into our columns headers, we'll just replace those with our double X's. And now we've created our table shell. I can't remember if I said this out loud or just replied in the chat, but there was a question about, what do we do if we don't have our data yet? And in that case, we would create dummy data. And I've linked to the article on our package down website where you can see more information about how to create these shells with both uniform and custom statistics. And that's going to take us to our last section about exporting.
Okay, it is great to create interactive tables in R, but obviously the point is we got to get these tables out of R and into the hands of someone who needs to review them. So I think one of the most powerful features of gtreg is the exporting options that come with it. So we are going to talk today about this very bottom box here, where we can get our results in HTML, Word, PDF, and Excel. So we've kind of put our favorites here as when you want HTML results. I think GT is the perfect engine to create those tables. If you need Word, my preference is Flex Table. PDF, I love Cable Extra. And for Excel, I use Hux Table to export to Excel. So let's go over a few of those options and see what we're working with. So before we get into the weeds here, I just want to give you a quick view at a nice little table. Let's see. I'm trying to get to my table, but all right. So here you can see the functions as GT, as Flex Table, as Hux Table, as Cable Extra, as Cable, and as Tibble. And all of these functions will take a gtreg table and convert it to that type. And once it's that type, you can again use all of the formatting functions from that type of, from that class of table. So you can see here what output types are supported by each of the various engines here, and why we've selected, GT is our favorite HTML, and for example, Flex Table is our favorite Word. Pretty soon, I think GT is going to be available and working perfectly for all output types, our RTF, Word, PDF, and HTML. They're already very, very close. So I would expect to see that in the next month or two, I would guess. So if you like GT, like I love GT, then that could be a good option just to stick with GT going forward after those small modifications have been made to make those tables perfect for RTF and Word as well. So let's jump back to our slides. All right, so again, the table you've already seen before, a lovely adverse event summary table. We're saving it as table 1, and we are piping in our DF adverse event to TBL AE. We've seen this all before, and we're bolding our labels. By default, GT is the print engine, gtreg, the package is written as a companion to the GT package. So here is our default HTML GT print engine table, and it's what you've seen already. It looks great. Here, we are taking that table 1 object and we are using as flex table to export it to a class flex table. And it largely looks the same here, and I think that's a really great option for being able to export to various output types. Here's an example of Excel via the Hux table package. Again, largely all the formatting here has been preserved, and the results look great if you need your results in Excel files. And lastly, PDF, here's an example using Cable Extra. Cable Extra, it's been around for quite a while, has a lot of really great features and a lot of great documentation on how to make, additional changes here. So you can see here, as we converted to Cable Extra with as Cable Extra, we use the book tabs equals true argument to modify some default styling of that PDF table. And we've also piped it into Cable Extra cable styling to give some gray stripes. So lots of options there as well. So it's super powerful to be able to export your table to like any output type that's supported by the 4, 5, 6 most popular tabling packages in the R ecosystem. So I love it. So that's going to bring us to our last exercise today, exercise 6.
Yeah, so have your hand exporting one of the tables. I believe that says your table shell, but you can choose any of your favorite tables to Word or Excel, because those are the quickest to do, but you're also welcome to fiddle around with R Markdown to PDF format in the 5 minutes that you have if you want to start that timer, Daniel. All right, and so our last exercise is about getting these tables out of R when you need to. See if I can find my RStudio Cloud instance. Here we go. And so we are exporting our T2 shell as either Word or Excel just for simplicity here. So I'm going to take my T2 shell that I've already created, pipe it into as HuxXLX. I'll note that if I've already installed a few packages on this RStudio Cloud instance for you. If you are working on your own computer, you'll need the Hux table package in order to execute this, I believe. And so I think this finished executing. So here we have our summary shell as a XLSX file. We can take a look at that. And it opened in a new tab for me. There you go. So you might need to do a little bit of manipulation to see all of the headers and all of that, get the spacing shown quite right. And if you want to see it in Word, we can also do T2 shell to as flex table, again, requiring the flex table package if you don't have it installed already. We'll save as docx. And we'll do summary shell dot docx. Not fine. I thought I installed it. Let's see here. Yes, I did install it. And you have to specify the path equals as well. There we go. So there's our summary shell docx and a Word document as well. Okay, are there any questions? I think I did see some questions in the chat about exporting to RTF. And like Daniel discussed, RTF is supported in Huxtable now, but you can wait on anticipate changes in GT that will make exporting to RTF smoother straight from GT as well. Are there any other questions that need to be addressed in the chat? All right. I really want to give you all a big thank you for hanging in there until the end of this workshop, especially with like me coughing. I know that wasn't very pleasant, and Daniel picking it up so quickly. So thank you all for joining us today. We would love to get your feedback on this workshop, both in terms of the workshop content and format and the gtreg package. So there is a link that is going to get posted in the Zoom chat in case you can't access the slide. And we would love your feedback on this workshop. So please take the remaining 5 minutes to provide that feedback. We do have our website for gtreg, a package down website with some long form documentation about a lot of these things that you had questions about on table shells and modifying tables. The gtreg package is on CRAN, so you can install it from CRAN, or you can install the development version from GitHub. If you want to keep in touch, we recommend doing this on GitHub. So if you have any issues or bugs to report, feel free to file an issue for us. And if you have features requests or you just want to know if things are possible, please post that in the discussions. So thank you again so much for joining us today. We hope it was worth your time.